Good morning, my friend. Good morning, good afternoon, good evening, my friends. I welcome you all to Part 2 TCT 2020 summary. Today we are going to review CAR-T, GVHD, and gene therapy presentation. Lots of good abstract. We'll try to provide you pertinent brief information on selected clinical abstract. Abstract not selected for presentation or discussion today are equally high quality, but we can't discuss everything in 55 minutes. Again, I have no conflict of interest to discuss. I'm going to present abstract in a chronological order, starting from Wednesday presentation to Sunday, ending with a late-breaking abstract. Outpatient treatment with Lysocell in 3 ongoing clinical studies in relapsed refractory B-cell non-Hodgkin's lymphoma. This study was presented by our friend Carlos Bashir. He's our neighbor across the street. We thank him and investigator for sharing his presentation. Making sure I'm loud and clear. Yep. Lysocell is a BMS product, previously Celgene and originally Juno. FDA has granted priority review to Lysocell in relapsed refractory large B-cell lymphoma in February 2020. Company seeking approval of anti-CD19 CAR-T therapy Lysocell for the treatment of adult patients with relapsed refractory large B-cell lymphoma under priority review after at least 2 lines of therapy. Expect we will have another commercial CAR-T product soon. I'm not going to go into the background information in detail. As we all know, CAR-T cell therapy is not easy treatment. It required lots of monitoring, generally needing inpatient management. Most patients treated in US with relapsed refractory large B-cell lymphoma receive therapy at non-academic or non-university center, community hospital, where they have the best outpatient program, in and out. And if we do CAR-T therapy in an outpatient setting may lead to wider utilization and improved access. Author presented data on safety and efficacy of CAR-T in patients with relapsed refractory large B-cell lymphoma who were monitored as an outpatient at both university and non-university sites in 3 separate studies. As we all know, Lysocell is a CD19 interaction 4-1BB CAR-T product administered at equal target dose of CD8 positive and CD4 positive CAR-T cell. Across the Lysocell clinical program, treatment in the outpatient setting is allowed per investigator's discretion and followed standard guidelines for safety monitoring and adverse event management. Slide outlines the study design of all 3 Lysocell clinical trials: Transcend NHL 001, Outreach, and Pilot Study. Transcend NHL 001, a multi-center phase 1 study of Lysocell in relapsed refractory large B-cell lymphoma, was presented in the late-breaking abstract also, and I'm going to review soon. If you can look at the patient's eligibility criteria in the middle and left box, patients with mild to moderate organ dysfunction and secondary CNS lymphoma were eligible for the study. Very interesting. Most clinical trials exclude those mild to moderate organ dysfunction. Slide summarizes the sites and patients' requirement for outpatient monitoring. Site requirements were BMT program, phase 1 trial capability of program, outpatient infusion center, affiliated pharesis center, multidisciplinary medical team. Hospital requirements were capable of CAR-T adverse event monitoring and management, staff trained to manage CAR-T cell toxicity, also caregiver support for the first 30 days after Lysocell infusion. In short, I would say kind of FACT or JC accredited IEC program. So most important message here is after initial observation on day of Lysocell infusion in outpatient setting, Patient will be released with caregiver educated to recognize early side effect of early signs of CRS and neurological events. Daily monitoring, daily for at least first 7 days, clinic visit or phone call, then at least two times a week during first month. In our program, first few days we do in-person visit and lab investigation in the morning, and evening we do telephone visit. Hospital admission at first sign of fever, CRS, or any neurological events, or any other therapy or disease-related complication. Slide summarizes patient characteristic across 3 lysocell clinical program. Total 44 patients treated in the outpatient compared to 269 in an outpatient treated in Transcend NHL 01 clinical trial. No major differences between both groups. In terms of disease characteristics, 2/3 of patients had chemorefractory disease, and more than 1/3 of patients had prior auto in each group. Major grade 3 or more treatment emergent adverse events and were mostly pancytopenia as expected. Very few patients had grade 3 or more CRS. No grade 4 treatment emergent adverse events were reported for patient monitored as an outpatient. Slide summarizes adverse event of special interest in outpatient. Overall, very few patients develop more than or equal to grade 3 CRS, only 5% patient in outpatient group. Neurological events, this is very impressive. Again, any grade was observed in 30%. Again, only 5% of patients develop more than equal to grade 3 CRS, neurotoxicity, 10% in the inpatient group. Tocilizumab or corticosteroid for CRS in only 11% in outpatient group. Very impressive, right? Half of the patients were not hospitalized among outpatient group. Very impressive. Meaning, group who started CAR-T infusion as an outpatient remain outpatient, half of them. Median length of stay among outpatient group was 6.5 days once they required hospitalization versus 11 days in the other group. Median time to hospitalization after CAR-T infusion as an outpatient was 5 days, but only 20% patient required hospitalization before study day 4 plus 4. This is great in terms of reimbursement purpose in United States because they want to be outpatient at least 72 hours after the infusion to get good reimbursement as an outpatient. No increase in ICU admission compared to inpatient, although very few patients require ICU admission, overall 5% in the outpatient setting and 7% in the inpatient group. We wanted to know efficacy, right? Efficacy outcomes are very important. We don't want to compromise the outcome by treating patient outpatient. Overall response rate was 80% among outpatient group and 73% in the inpatient group. More than half of the patient achieved CR in both groups. In summary, outpatient lysocell was successfully implemented in multiple clinical trials at both university and non-university sites. Almost half of the patient did not require any hospitalization after lysosol. No increase in ICU level care observed, and no compromise in outpatient outcome. Most fascinating to me was 45% had CRS and neurological events. That's okay because very few patients had grade 3 or higher neurological event or CRS. And the most important other part was median time to onset for CRS or neurological events was late. Median time to development of CRS was 5 days, and median time for neurological event was 8 days. So that means we can do most early part as an outpatient. Our next presentation is acute graft versus host disease is less severe and associated with lower non-relapse mortality after haploidentical transplantation with PTCY. Presented by our well-wisher, Dr. Rima Saliba from MD Anderson Cancer Center on behalf of GVHD Working Committee CIBMTR. We thank Dr. Saliba for providing her presentation for today's educational discussion and share the quality work done by CIBMTR with our international friends. I don't think we need to review the PTCY literature, well known to us. PTCY has changed the epidemiology of GVHD. I would say we don't see much severe acute GVHD nowadays. This is my personal bias, but this credit goes to PTCY. And if patient develop GVHD, we do have lots of options nowadays, and we strike early. PTCY mechanism in GVHD pathophysiology are still evolving. So although some may not agree with that. How PTCY affect the acute GVHD, clinical manifestation and prognosis remains largely unknown to me. Our main question in these studies are, and generally this is what we have a question, in patient with acute GVHD, are the clinical manifestations and prognosis differ after PTCY versus conventional GVHD prophylaxis? Example, tacrolimus MMF or tacrolimus methotrexate. Second most important question is, are differences consistent between older patient and younger patient? Very important because we have more and more older patient receiving allotransplantation. Valid question, right? And we want to know the answer because we want to use PTCY now in almost every allotransplant procedure. Lots of data. Match-related donor with PTCY, MERD with PTCY, mismatch unrelated with PTCY. Hope not after cord blood transplant. I don't know any literature on CBT using PTCY. So in this study, author hypothesized that less severe manifestations and better prognosis after PTCY-based GVHD prophylaxis regimen. Slide summarize the study population. Allotransplant period from 2013 to 17, recent PTCY cohort. 264 of 758 patients received haplo with PTCY and MMF-based GVHD prophylaxis regimen, most popular haplo regimen across the globe. And this group was compared with 1,163 patients receiving MERD transplantation with conventional GVHD prophylaxis, mostly with tacrolimus. Standard study endpoints. Author wanted to know characteristic of acute GVHD, organ distribution and severity, and authors also wanted to know prognosis after developing acute GVHD. Valid question, right? Non-relapse mortality and risk of chronic GVHD in patient developing prior acute GVHD. Standard statistical methods were used. Important thing was additional subset analysis was performed in this study for patients who were 60 or older. Very important to know about the older cohort. As expected, haplo donors were older than match unrelated donor. There were more female donor to male recipient in haplo group, including in older cohort also. As we all know, older donor female donor for male recipients considered high risk for GVHD in many studies. And this study in haplo group had both adverse factors in excess number compared to other group. More acute myeloleukemia patients in haplo group and MDS in MERD group. Similarly, as expected, more bone marrow graft among haplo group, and majority of MERD recipients received peripheral blood stem cell graft. Severe acute GVHD grade 3-4 was significantly lower in PTCY haplo group, 28% versus 38% in MERD recipients. 6-month cumulative incidence of grade 2 to 4 acute GVHD was significantly lower in haplo group. Same results were seen when older patients analyzed separately. 25% patients developed severe acute GVHD grade 3-4 in PTCY group versus 41% grade 3-4 severe acute GVHD in MERD group. Multi-organ involvement was less common in PTCY group compared to MUD group. More than 2 organ involvement was seen in 26% patient in haplogroup versus 35% in MUD group. Severe grade 3-4 lower GI GVHD and liver acute GVHD grade 3-4 summarized in the bottom table. Both type of GVHD, severe lower GI and severe liver GVHD, were significantly less in PTCY haplogroup versus MUD group. Similar findings were seen in when older patients analyzed separately. Severe lower GI GVHD in 12% in haplogroup, almost double in MUD group, 23%. Very difficult group to manage, right? Severe lower GI in older adults, in almost quarter of patients age 60 and above receiving MUD transplant, worst possible scenario, pain in the neck. Widely separated graphs curves here significantly decrease risk of non-relapse mortality in patient with acute GVHD in haplogroup compared to MUD patient developing acute graft versus host disease. Benefit of PTCY haplo in reducing non-relapse mortality was not seen in patient with female donor for male recipient haplogroup. This is very important and interesting finding. Risk of NRM was no different between MUD or haplogroup, but haplogroup with male donor for female donor for male recipient. In multivariate analysis, grade 2 to 4 acute GVHD in haplogroup was associated with significantly lower non-relapse mortality, but this effect was limited to donor recipient pair that were not sex match, means not female to male. In sex mismatch pair, female donor for male recipient in haplo, non-relapse mortality was comparable between haplo and MUD, and this is again interesting finding. Effect of gender mismatch was profound when older cohort was analyzed separately. You can see multivariate analysis table with hazard ratio, excellent outcome. Now, what about risk of chronic GVHD in patient developing prior acute GVHD? In multivariate analysis, summarized here in the table, risk of chronic GVHD was higher in MUD group compared to haplo, but only in patients receiving non-ablative MUD transplantation. Similar results were obtained in older cohort. You can see here, risk of chronic GVHD was higher in MUD group compared to haplo, but only in patients receiving non-ablative MUD transplantation. You can see here in table, top rows. Organ involvement did not differ between two groups. However, stage 4 was less frequent in haplogroup. This difference was most pronounced for lower GI and liver acute GVHD, less in haplogroup. Bottom line message from this large pivotal study, acute GVHD after haplo plus PTCY, less upper GI involvement, less severe GI, liver and lower GI organ involvement, lower non-relapse mortality except for female to male donor recipient pair, lower chronic GVHD except in recipient of myeloablative conditioning regimen. This is our next abstract, very interesting abstract presented by Yanni from Memorial cord blood transplant group. I know we don't do much cord blood transplantation nowadays, but this study has a great clinical implication overall in our transplant practice. It was presented as an oral presentation. Just briefly background, cord blood transplant extend transplantation to those who don't have suitable related match or unrelated donor. Cord blood transplant without ATG, those who were doing cord blood transplant before, I'm sure remember, without ATG provides comparable transplant outcome, but with associated low chronic GVHD and robust graft versus leukemia effect. However, ATG-free cord blood transplant is associated with high pre-engraftment syndrome and severe acute GVHD. We have also published our own data. At the same time, ATG-based cord blood transplant associated with impaired immune recovery, lots of infection, a lot. I'm sure you remember all viral infection. Recall our old days several years ago. Therefore, what we need is enhanced but non-lymphodepleting, not lymphodepleting GVHD prophylaxis strategies required. Nice background. Slide summarizes the rationale for IL-6 receptor blockade for prevention of acute GVHD and pre-engraftment syndrome. High IL-6 level after adult donor hematopoietic cell transplantation reported to be associated with incidence of acute GVHD presented in last ASH meeting. And also IL-6 level after cord blood transplantation reported to be associated with pre-engraftment syndrome. All nice references are listed here. This is an Australian randomized trial presented at the ASH meeting by Professor Hill and team, showed some promising result. 10 to 15% reduced risk of acute GVHD using TOSI, although not statistically significant, but important study. So in the present study, author hypothesized that addition of TOSI to cyclosporin MMF-based double cord blood transplant will reduce the incidence and severity of acute GVHD and pre-engraftment syndrome without compromising patient's outcome, without compromising GVL effect. To test this hypothesis, this is a phase 2 clinical trial design using the reduced toxicity. Some will say this is a myeloablative conditioning regimen with thiotepa and 400 TBI, followed by double cord blood transplant, standard GVHD prophylaxis with cyclosporin mycophenolate, and study drug, single dose of TOSI on day -1, similar to phase 2, phase 3 trial presented before in adult donor transplant recipient. Study design summarized here. Primary endpoint, day 100 acute GVHD with aim of less than or equal to 40%, from around 65% in historical control after double cord transplantation, meaning 50% reduction is the goal. Highly ambitious plan, right? Let me inform you, primary endpoint was not met, but there was no safety concern due to improvement in key secondary endpoint, and author extended sample size to better understand the efficacy endpoint, and clinical trial is still ongoing. Standard eligibility criteria, everything what we all consider in our program, nothing unusual, not going to go in detail. Disease categories included all heme malignancy except myelofibrosis as expected. As expected, a disease myelofibrosis will try to avoid using cord blood transplant for an obvious reason. Author presented information on 33 patients, all with 100-day follow-up. Median age of cohort 48 years, median weight 79 kilo. Majority, almost 80% of patients had acute leukemia diagnosis. Reasonable cell dose, median nucleated cell dose 2.31 into 10 to the 7 kilogram per unit. Highly reasonable cell dose. 31 of 33 patients engrafted WBC. One patient had a graft failure in the setting of low graft viability, disseminated infection, and second patient had early transplant-related mortality with incomplete count recovery. Not bad. This is very impressive. This is a major finding. Author reports significant reduction in pre-engraftment syndrome and febrile neutropenia. 70% patient had no pre-engraftment syndrome. Look at here, pie graph. And very importantly, 60% had no febrile neutropenia. Additionally, those 10% who develop pre-engraftment syndrome only half required systemic steroid. Only fraction of all patients did not require required steroid. Only 5 out of 33. Study data showed very low incidence of severe grade 3-4 acute GVHD. The day 100 cumulative incidence of grade 3-4 severe acute GVHD was only 4%. Figure is a little bit confusing to me here because on top blue line it says grade 2 to 3 and bottom red line says grade 3. But I know I was there for presentation. It was very low. Very low incidence of severity of lower GI acute GVHD. This is again very important. Very fascinating because severe lower GI GVHD, as we all know, is pain in the neck. It is still considered, I would say, our major weakness to manage. Very low incidence. 1 year TRM was only 17%. Of 5 patients who died from TRM, 3 with documented viral infection is still a problem after cord blood transplantation. Importantly, no acute GVHD related death. 1 year relapse rate was only 11%, meaning, and this is important to me, TOSI preserved the GVL effect. With a median for survivors follow-up of 15 months, 1 year overall survival was 82%. Impressive result. Progression-free survival 71%. Look at the GRFS 69%. GRFS approximate progression-free survival, meaning those patient progression-free remain GVHD-free. Age-adjusted comorbidity index had no impact on progression-free survival. Very good. In summary, and very important message from this paper was marked reduction in grade 3-4 acute GVHD and less lower GI involvement using TOSI-based cord blood transplantation. Marked reduction in incidence of severity of pre-engraftment syndrome. Investigation of long-term outcome is still ongoing. Our next abstract, safety of autologous hematopoietic stem cell transplantation with gene therapy for transfusion-dependent beta thalassemia, sickle cell, and cerebral adrenoleukodystrophy, ALD. One of the top class abstract presented by our well-wisher, Dr. Walters. On behalf of his team, want to thank him and his international well-known team leader, and also thank Bluebird Bio for sharing this presentation. Let me inform that there was a similar class of presentation, abstract 47, just before this abstract, presented by Professor Paul Orchard from University of Minnesota on lenti-D hematopoietic stem cell gene therapy in cerebral ALD. Again, thank him, thank his team, and Bluebird Bio for these slides. I will try to give you all snapshot of both abstracts in this short summary. As we all know, established curative treatment for non-malignant genetic diseases, allotransplantation. Overall survival of these 3 diseases after allotransplant summarized in the slide, well known to us, not going to go into the detail. What we can say here is that outcome is very good in HLA identical sibling, patients who are young and early in their disease. Survival is worse for greater disease burden, patient receiving alternative donor transplant. So there is an opportunity to improve outcome in those with gene therapy, right? Autologous hematopoietic stem cell gene addition does not have some of these limitations like we see in after allotransplantation and may have a similar efficacy with improved safety profile. Let's look at the data. Ex vivo gene addition therapy using lentiviral vector was evaluated in patients with transfusion-dependent beta thalassemia using this long name. I can't pronounce it, Betty Belger. Thank God we do have a short, easy abbreviated name, Betty cell. I still don't know why company is always difficult name, and all CAR-T products have a very difficult name. I don't think I can pronounce this Betty correctly. Study numbers are highlighted here in red. For sickle cell disease using lentiglobule and cerebral ALD using lenti-D in ALD 102 study. Of total 115 patients across this study, 51 patients with more than 2 years follow-up. Stem cell collection began with standard methodology. These cells were shipped to the central manufacturing facility where the lentiviral vector transduction occurred, and those product satisfied to release were cryopreserved and sent back to the transplant center where patients received myeloablative conditioning with BCMA followed by product infusion. Product thawing occurred at the bedside. Then 2-year follow-up with the option to continue participate in the study, 13-year follow-up. I wanted to inform you here that an ongoing study, ALD 104, is investigating a different myeloablative conditioning regimen, FluView. I guess many of us will appreciate these changes. Slide summarize the patients and treatment characteristic. Thalassemia and sickle cell patient initially targeted the adults and later extended to the children. Cerebral ALD study involves boys with early signs of progression as expected. All thalassemia and sickle cell patients were in advanced disease. You can see here, all patients had a myeloablative busulfan-based conditioning regimen. AUC is summarized here in all three across 3 studies. Mobilization and apheresis toxicity profile shown here, mostly related to thrombocytopenia, leukocytosis, electrolyte imbalance, which is associated with pheresis. Nothing unusual. Among sickle cell patients, vaso-occlusive pain was common adverse event and it occurred in few patients. Adverse events related to conditioning summarized here, with most common toxicity were mucositis, febrile neutropenia, and other common events after we usually see after myeloablative conditioning regimen. 9% of patients with thalassemia developed serious VOD. All resolved following treatment with defibrotide. No patient with sickle cell disease or cerebral ALD had a VOD. All the patient engrafted, no graft failure. You can see here ALD patient engrafted fastest and thalassemia slowest as expected in most thalassemia series. Patient platelet engraftment took longer than we typically see after peripheral blood allograft. Some of the adverse events related to bleeding in patients with thalassemia was due to delayed platelet engraftment, but none of the events were life-threatening. Median hospitalization duration from starting conditioning to discharge summarized here, about 4 to 6 range, so to 6 weeks range. Not unexpected. Remember, these are all clinical trial patients, and we and study sponsor usually want to keep them safe. No drug product related death. One patient with ALD had early and rapid progression while on study and died. One case was monosomy. 7 MDS reported 36 months after infusion in sickle cell patient. This was not related to lentivirus mediated based on their further investigation on viral copy number in BAST cell. And it was ruled out this is not related to study drug, and it was presumed related to busulfan. Adverse event attributed to the drug product summarized here. One event of thrombocytopenia in thalassemia and BK-mediated viral syncytial in ALD were serious. Majority of drug product related events were non-serious. Here for your reference, in case you want to review later, this is all superb to me. Figures showed stability of vector copy number detected in peripheral blood over time through 5-year post-infusion period. And this is associated with clinical outcome. In thalassemia, Look at these numbers. 90% of patients with more than 6-month follow-up became transfusion-free. Among cerebral ALD, 88% of evaluable patients were alive and had no major disability at month 24. Among sickle cell disease patients, markedly reduced vaso-occlusive crisis and acute chest syndrome in patients with higher viral copy number, vector copy number after refined manufacturing. All 3 abstracts were presented in the meeting. You can review in details later. To me, this is a really amazing and solid result. Bold circles are in the patient with refined manufacturing in this graph with higher vector copy number, which is a recent cohort. In summary, there was no graft failure, no drug product related death in 115 patients across 3 studies, 3 different diseases with up to 5 years follow-up, 51 patients with more than 2 years follow-up. There was no insertional oncogenesis with 5-year follow-up. This was one of the issues. I think if you read the old literature in the past, old literature in 19th malignancy was the issue after gene therapy. So gene therapy for this hereditary non-malignant disease is acceptable alternative to allotransplant who lack suitable donor or patient more advanced age or those who are at high risk for allotransplant complication. Let me tell you all my friend, gene therapy is coming and soon in most cellular therapy program. Be ready, like we are ready for CAR-T almost everywhere for past several years. So next abstract is on risk adapted preemptive TOSI decreased severe CRS after CAR-T therapy in pediatric B-cell ALL. As we all know, patient with higher tumor burden at high risk of developing CRS after CAR-T, right? And one of the effective treatment for CRS is tocilizumab. However, TOSI in preventing CRS is largely unknown, not much data. Investigators in this study sought to determine the effectiveness of preemptive tocilizumab administration in decreasing the rate of grade 4 CRS in patients with high tumor burden. This is a pilot trial of risk-adapted prophylactic TOSI after TISA-cell. Again, nice abbreviation from long name. High tumor burden were defined as high tumor burden, classical definition with more than 40% blast, received preemptive TOSI for high persistent fever to temperature more than or equal to 38.5 in 24 hours. Primary endpoint, frequency of grade 4 CRS with observed rate of less than equal to 5 out of 15, predefined as a clinically meaningful. Comparator cohort with high tumor burden was receiving standard CRS management in their prior clinical trial. Reasonable. Preemptive TOSI was used in 15 patients compared with 26 patients with standard CRS with high tumor burden. All patients had grade 2 or more CRS, not unexpected. Grade 4 CRS was observed in 27% patients in preemptive TOSI group versus 50% in standard CRS, all with really high tumor burden. TOSI did not compromise CAR-T versus leukemia effect, with CR rate at day 28 was similar in both groups, 87% versus 85% in patients with high tumor burden in both groups. Very impressive CR rate in this population with more than 40% blast immediately before infusion. I don't think I need to read this conclusion side. You can review it later. Simple message is risk-adapted preemptive TOSI administration reduced grade 4 CRS, meeting the predefined study endpoint without impacting CR on day 28 in this high tumor burden population. Our next abstract is on multi-center retrospective analysis of outcome and toxicity with commercial axi-cell which is Yescarta, and Tisa-cel, also called Kymriah, for relapsed refractory aggressive B-cell lymphoma. I would like to thank Dr. Riddle and consortium leadership for sharing their presentation for me presentation today. I'm not going to go into the background here, very well known to us. FDA approved Axi-cel, Yescarta in October 2017, Tisa-cel, Kymriah in May 2018 for relapsed refractory aggressive B-cell lymphoma. Objective of this study was to describe the efficacy, safety, pattern of use of this product in a center that had option to prescribe both product, commercial Yescarta or Kymriah. 170 patients were collected for Axi-cel, Yescarta, and 158 infused. 94 patients collected Tisa-cel, Kymriah, and 86 were infused. More older patients who received Tisa-cel, Kymriah, more patients with primary refractory disease that received Axi-cel, Yescarta product. Tisa-cel patient group were more heavily pretreated, 80% with 3 or more regimen. Tisa-cel group had lower comorbidity index. Tisa-cel is again Kymriah. Tisa-cel group more likely to receive bridging chemotherapy versus Yescarta product Axi-cel. Out of specification was seen in almost 1/4 of Tisa-cel Kymriah group, with majority of being viability in range of 70–80%. Generally, centers are okay with 80%, up around 80% viability. Almost half of the patients treated in Axi-cel group were ineligible for original JUMA-1 study, and about 30% of Tisa-cel group were ineligible for original Juliet trial, the respective pivotal trial. This is what happens after product get approved, right? Real case scenario, total safety taken into place when we do the clinical trial. And once product is approved, we ignore some of the comorbidities and use like we have used in this clinical presentation. Authors were able to harmonize the reporting of recent ASTCT toxicity grading for better comparison. ASTCT toxicity grading was published in BPMT in April 2019. In terms of CRS, 85% had any grade CRS, 8% grade 3 or more after Axi-cel, Yescarta product. 41% had any grade and only 1% had grade 3 or more CRS after Tisa-cel, which is a Kymriah product. Looking at the neurotoxicity, most importantly, 33% of patients had grade 3 or higher neurotoxicity after Axi-cel and none in Tisa-cel recipients. Look at some of the toxicity management. TOSI was used in 61% patient treated with Axi-cel and 15% patient treated with Tisa-cel. Similarly, steroid use was significantly higher in patient treated with Axi-cel compared to patient who received Tisa-cel product, 53% versus only 8% in Kymriah product. Additionally, Axi-cel product population, 6% of patients required their additional anti-cytokine therapy, especially saltuximab. Looking at the grade 5 adverse event, meaning non-relapse mortality, 9% in Axi-cel recipient versus 2% in patient receiving commercial Tisa-cel product. Non-relapse mortality, most early deaths less than 30 days were treated related to severe neurotoxicity in Axi-cel group versus 0% NRM in first 30 days in Tisa-cel group. Looking further, death beyond 30 days, unrelated to lymphoma progression, 7% in Axi-cel, 2% in Tisa-cel, mostly infectious complication. Let's look at the efficacy, more important, right? Median overall response at day 90 shown here, generally in line with respective pivotal clinical trial, meaning clinical trial product and commercial products were equally effective as expected. This is a Kaplan-Meier curve for survival analysis in patients receiving exa-cel product. Median progression-free survival of 6.7 months from cell infusion, decent outcome in relapsed refractory diffuse large B-cell lymphoma. Looking further into patient treated with TCT, median progression-free survival of 3 to 3.2 months. Authors were also able to look at the number of resource utilization metrics in patients treated in this analysis. 92% of exa-cel product received in context with inpatient versus 37% among T-cell product. Median hospitalization days within 20 days of infusion was 16 days in patient receiving Yescarta product versus 2 days in Kymriah product. ICU transfer was more common in same patient with exa-cel Yescarta product. In conclusion, T-cell recipients were more likely to be older, more heavily pretreated, and to receive bridging therapy. T-cell appeared to be associated with a lower incidence and severity of CRS and less neurotoxicity. Efficacy outcome in the commercial setting appeared to be similar to pivotal trial. exa-cel was associated with longer hospital stay, increased incidence of ICU transfer, and greater use of TOCI and steroid. But important to know is efficacy. That's what we want to know. Difference between the product. Author did mention that propensity score matching will be needed to more adequately compare efficacy outcome among both products. This is what we wanted to know. In our program, again, maybe biased, we try to use Yescarta exa-cel product in young but super bad disease and also in patients where manufacturing failure might occur with Kymriah product. Again, this is a biased opinion. Nice abstract. Next one, presented by Gunjan, our well-wisher from Memorial. Medicare patient receiving CAR-T therapy, a first real-world experience at patient characteristic, healthcare utilization, and cost. This cost our main question, right? Always right. How much it will cost? First question comes into mind, especially from the administrator of the hospital. Approximately 74,000 Americans with NHL each year, approximately 1/3 of them with diffuse large B-cell lymphoma. And many of these patients are Medicare eligible, and these are many our respected older adult seniors. Objective of this study is to describe the demographic and clinical characteristic of Medicare patient receiving exa-cel or TCT, and to compare healthcare utilization, cost, and outcome pre and post CAR-T cell therapy. Important aspect of this study was that study provides the first real-world look at patients' CAR-T experience using Medicare fee-for-service claim after FDA approval. This is very important for CAR-T program in United States. Data source was CMS with patient having 100% Medicare fee-for-service and only Part A and Part 2, Part B claims. Part D data were not available at the time of submission of abstract. Patient received CAR-T cell therapy from October 2017 to September 2018, and this is called index period. Authors ended up with 2007 patients who met the criteria with median age of 71 years, ranging from 34 to 83 years. So 92% of patients had ICD code C83.3, which is diffuse large B-cell lymphoma. Other 8% had other high-grade lymphoma. 45% were treated in CAR-T clinical trial. Majority of patients had Charleston comorbidity index of 3 or higher, in 80% as expected in median age of 70, and that's why many of these were not eligible for pivotal clinical trial. Classical lymphodepleting regimen, No need to go into the details. Median follow-up 12 months. At 6 months, 73% of patients were alive. Median overall survival not reached yet at the end of this study. Now, the cost is important. This is how it was calculated. This will be too complicated for European audience, but let me briefly summarize. When the author looked at the actual CAR-T episode, actual time in the hospital around the infusion, in the outpatient setting, around the infusion. This cost is the allowed amount actually paid by the Medicare to the hospital. Authors were able to show that prospective payment system exempt cancer hospital had a significantly lower payment compared to non-PPS exempt hospital for that actual CAR episode, real CAR-T episode. Highest mean cost of CAR was in outpatient testing. And this is very important, I think, because high reimbursement rate in outpatient setting. And that's what all CAR-T program in United States like to start with CAR-T infusion as an outpatient for the better reimbursement purposes. Now, the cost outside the index period, as I mentioned before, index period was between October 2017 to September 2018 when CAR-T was administered. Pre-index and post-index, 6 months before and 6 months after, significant reduction in the cost of management outside the index period. So in summary of this complicated abstract, and very important for the reimbursement purpose for the administrator, they said older patient with multiple comorbidity had a they were treated successfully with CAR-T. Post-CAR care was associated with fewer hospitalization, means 6 months after CAR recovery, short time in the hospital, fewer AD visit, and lower health utilization care management. Now, I wanted to thank Dr. Levine, one of the lead senior author, and Rishikesh for providing me PowerPoint slide for this today's presentation, Magic Algorithm Probability, commonly referred as a MAP, a novel laboratory biomarker for response to treatment of acute GVHD. Very important prospective multi-center international study. Our program is one of the participating center. Dr. Kitko, director of our pediatric program, is our institutional PI. Generally speaking, acute GVHD is our top class enemy for past several decades. Clinical response to treatment at day 28 is widely used clinical trial endpoint because it predicts non-relapse mortality. Recently, several serum biomarkers have been validated to predict the long-term outcome at single time point. However, there is no response biomarker for GVHD treatment. Very interesting study, my friend. Mount Sinai Acute GVHD International Consortium, MAGIC. In US, we are very good at abbreviation. MAGIC, the group of 20 international centers collect high quality clinical data and longitudinal serum sample. Author used the MAGIC database to study the serum level of ST2 and Reg3 alpha, which reflect the crypt destruction that is responsible for vast majority of non-relapse mortality. Author quantitate the crypt destruction in the map, which is the individual's estimated probability of 6-month non-relapse mortality based on their serum biomarker concentration shown in the equation. This is a very complicated formula I don't understand. In this study, author hypothesized that MAP, which is magic algorithm probability, after 28 days of treatment would predict long-term non-relapse mortality and function as a response biomarker. Amazing, right? Study summarizes the study design. All patients treated with systemic therapy for acute GVHD and who had both clinical and serum sample available at start of treatment and after 20 days. divided into two cohorts, treating cohort and validation cohort based on timing of treatment. Treating cohort was used to develop model and validation cohort was used to test the model. Author first divided patient of their validation cohort based on their initial map into Ann Arbor group 1, 2, and 3. Ann Arbor group 1 had lowest MP map and Ann Arbor group 3 group had a highest map, more than 2.290 at the start of treatment. And this works very well. I'm the witness. Blue color meaning no 6-month NRM and red meaning 6-month NRM. Now those did have NRM had a higher or they had an increase in MAP after initial treatment compared to those who did not have NRM. Similar pattern in Ann Arbor 2 group. Among Ann Arbor 3 patients where baseline F was already high, survival was clustered among those with largest decrease in MAP, you can see here in blue line. The author asked whether MAP threshold crossing the above or below single, MAP threshold crossing to above and below single threshold after the treatment will predict long-term outcome. Yeah, we want one marker above or below. So author used MAP threshold 0.29 based on their previous study. Reference here in red, the through the machine learning method, which most centers use now. In the same threshold, it will distinguish Ann Arbor group 2 from 3 at the start of treatment. Shown here, Kaplan-Meier curve, significantly different survival outcome. Blue curve represent low MP MAP at the start of treatment and remain below threshold at day 28. 1 year survival 82% versus 48% because it was Ann Arbor group 1 with low baseline MP. So only 5% had above threshold at day 28. Among Ann Arbor 2, 27%, and Ann Arbor 3, 67% had MAP increase above the threshold at day 28 and proportionately poor outcome. Impressive data. MAP predicted non-relapse mortality in both clinical responders and non-responders. High MAP among clinic responder, meaning patient responded clinically on day 28, but had high MAP at the same time. Only 10% but high non-relapse mortality, despite they had a clinical response at day 28. Majority of non-responder had low MAP, 58%, 57%, but had a low non-relapse mortality 1 year. I don't have blue dotted line here, little bit incomplete draft, but message is biomarker risk odds with their clinical response at day 28. Very fascinating. And this is very interesting. MAP significantly predicted non-relapse mortality than clinical response at the time of tested, shown here, black solid circle. As early as starting 1 week, mean MAP predicted response 3 weeks earlier than clinical response. In conclusion, MAP validated as a response biomarker for acute GVHD treatment. I don't think I need to read this conclusion side because this paper is already published. Slide. I think we are done with the timing, looks like. Yeah, sorry. This is published in Blood Advances. It looks like we are done with the time right now. I have several more presentation for the part 3 of our TCT highlights. I thank you very much for your attention, and I'll be happy to answer any few questions for remaining 2 minutes. Hello? Arnold? So let's continue with our few more abstract, very interesting abstract I wanted to show you. This is another IAC abstract, long-term outcome of patient with EBV-driven PT-LD following solid organ transplant or liquid allotransplant treated with TAB cell in multi-center expanded access program study or our institute participation study. Very interesting study. This is very well known that patient undergoing solid organ transplant or liquid allotransplant are at risk of developing EBV-driven PT-LD. Problem starts when patient fails rituximab, right? With or without chemotherapy, and outcome is very poor for those patients refractory PT-LD. Tab-cel is an investigational, off-the-shelf, allogenic T-cell immunotherapy generated from healthy donor that is selected for given patient from existing library based on HLA restriction and matching. So fascinating. Tab-cel is produced by Atera Pharmaceuticals. Author reported long-term outcome of tab-cel multi-center expanded access program study in patients with relapsed refractory EBV-related PT-LD following allotransplant in 14 and solid organ transplant in 12 patients. Tab-cel matched by more than or equal to 2 out of 10 HLA alleles, including more than or equal to 1 HLA alleles through which tab cell exerts cytotoxicity. Easy. In short, most patients will find tab cell off shelf if there is an indication to use it. Administration was 5-week cycle intravenously on day 1, 8, 15 with an imaging based on response assessment around day 28. All patients who received tab-cel had a prior rituximab. Most solid organ transplant recipients also had a prior chemotherapy. Most patients with intermediate or high-risk PT-LD IPI, no tab-cel related adverse event led to treatment discontinuation or death. Decent outcome in this worst possible scenario, 2-year overall survival, 79% in allotransplant, 80% in solid organ transplant recipient. Think about refractory PT-LD in a haplo patient or after MUD on high dose steroid for GVHD with PT-LD. Rare scenario, but think how many patients you expect to survive this complication. This is the best outcome of relapse refractory EBV positive PT-LD I have ever read or reviewed in my short career. The favorable 2-year survival in this predominantly high-risk population, for whom there is a no approved alternative therapy. Our next presentation is CAR-T therapy in patients with relapsed refractory mantle cell lymphoma, result of phase 2 ZUMA-2 study. Now, study was presented in the best abstract session, abstract number 1, but I'm not going to go into detail because study is already published in New England Journal of Medicine on April 2nd of this month. As we all know, outcome of relapsed refractory mantle cell lymphoma is extremely poor. ZUMA-2 clinical trial investigators presented this interim efficacy and safety result of ZUMA-2 trial. All patients with relapsed refractory mantle cell lymphoma, standard endpoint, safety and efficacy, and that's what we wanted to know. I'm going to go briefly review the updated result from the New England paper, not going to go any detail. CAR-T was manufactured for 71 patients with relapsed refractory mantle cell lymphoma and administered to 68 patients. 93% Of the 60 patients in the primary efficacy analysis had overall response rate, 67% had complete response. Look at the upper bound of confidence interval, 84%. Grade 3 or higher CRS in only 15% and neurological events in 31%. None were fatal. 2 grade 4 infection adverse event occurred. Good to remember, patient can still die from infection even after CAR-T therapy in current era. In short, CAR-T is very effective in patients with relapsed refractory mantle cell lymphoma. So I'd like to thank Tanya Jan for sharing this presentation. Hematopoietic recovery after CAR-T therapy for the hematological malignancy. We always talk about persistent pancytopenia after CAR-T cell therapy, and this study answers many of our questions. Propose prolonged cytopenia lasting several weeks to several months after CAR-T, extensively reported in clinical trial and observed in our clinical practice routinely. What we don't know, however, is that what is the timeline, pattern of hematological recovery, and what are the factors influencing this hematological recovery. And this study answers many of our questions. Authors started with identifying 115 patients who were treated with CAR-T after CAR-T therapy at Memorial. Of this, they excluded patients who had follow-up less than 30 days and those who relapsed or progressed or were given any additional treatment that can be potentially cytotoxic or myelosuppressive like chemotherapy, and conditioning for transplantation those who relapse. This to avoid confounding effect of cytopenia from this factor. Finally, 83 patients were included in the analysis. Of these, 30 patients were treated with Axi-cell, 10 with Tisa-cell for relapsed refractory NHL, both commercial product, 36 with CD1928 Zeta, homegrown CAR for relapsed refractory ALL, 6 with another homegrown BCMA-directed CAR-T for relapsed refractory myeloma. Of these 83 patients, CBC data was obtained for up to 12 months or until they were censored for the progression of disease or any additional things. This definition of recovery was used to indicate safe level without requiring transfusion or growth factor support, very reasonable definition. Hematological recovery, meaning hemoglobin more than 8, platelet more than 50, ANC more than 1000, WC more than 3000 without support. Complete count recovery means recovering of all 4 lines like in hematological recovery. Normalization of count, if count in normal range. And they use the ASTCT, IACH toxicity grading. Slide summarizes baseline data. Nearly half of the patient had lymphoma, 45% with ALL. More than 50% patient had more than 3 prior line of therapy. Grade 3-4 CRS was seen in 1/4 of the patient, and grade 3-4 ICANS neurotoxicity was in 36% of patients. This box plot shows united of all 4 lines. You can see here cytopenia did occur in almost all patients. This slide shows count recovery by percentage over time. All 83 patients are included at 1 month, while the number of evaluable patients declined over time. At 1 month of the 83 patients, majority had a recovered hemoglobin, 61%, platelet recovered, 51%, meaning more than 50,000. However, small percentage of patients had recovery of ANC, only 33%, and WBC only 28% at 1 month. At 3 months, complete count recovery in 47%, not necessarily normal count. Remember the definition, hematological recovery, complete recovery count, all 4 lines, and normalization of count. By 6 months, some degree, and more so by 12 months, most patients recovered all their count, but not necessarily normal. At 1 month, At 1 month, a rather small fraction had a recovered any or all of their count, dotted line. You can see to track the dotted line. Even as far as 9 months and at month 12, normalization seen in only 60 to 70% of the patients. Next, author conducted a univariate analysis to study if any patient or disease or CAR-T related factor associated with absence of complete count recovery. No impact of diagnosis or lymphodepleting chemotherapy between the group. Now, important thing is grade 3 or higher CRS, ICONS, neurotoxicity was significantly associated with no recovery in 97% of patients at 1 month. That means they were persistently pancytopenic. Authors were not able to construct many multivariate analysis due to the sample size and disproportionate number of events to create a traditional multivariate model. So author ended up doing Cochrane, Mantle, Hazel method to see the effect of high-grade CRS icons on effect of count recovery at 1 month after adjusting for CAR-T product or baseline cytopenia, which were significant factor in univariate analysis. So after this adjustment, absence of recovery of complete count at 1 month remains significantly associated with grade 3 or higher CRS and icons, respectively, p-value 0.02 and 0.01. You can see here in red highlighted. So not going to go details about conclusion. Main message in this presentation is grade 3 or higher CRS icons neurotoxicity is significantly associated with persistent pancytopenia beyond 1 year after transplantation in more than 90, up 1 month after CAR-T cell therapy in more than 95% of the patient. So this is one of the late breaking abstract presented on Sunday, last few abstract now, presented by Dr. Corey Cutler on behalf of Rockstar Study investigators. I'd like to thank Dr. Cutler and his team for sharing this presentation. Now, our institute participated in this study led by Dr. Jagasia, one of the leading GVHD figure and last author of this study presentation. I don't think I need to go into the details of pathophysiology of chronic GVHD today. Very complex, involves both B cell, T cell, and germinal center may play pivotal role in the setting up immunological milieu of chronic GVHD. As we all know, not online, this is what only I know. T cell mediated inflammation is the endpoint of acute GVHD, and fibrosis is often the final outcome of chronic GVHD. Row-associated coil kinase or ROCK kinase is a critical regulator of inflammation. ROCK2 inhibition selectively downregulate the pro-inflammatory Th17 cells and increase the regulatory T cell. You can see here a lot. Let me be frank with you. I don't understand much, but briefly, ROCK kinase system is also critical in regulating multiple pro-fibrotic process, and pan-ROCK inhibitor can prevent fibrosis in the experimental models. Selective ROCK2 inhibitor can also prevent collagen deposition, fibrosis, and scarring, and can be very useful agent in the chronic GVHD management. KDO25 is a novel, oral available, small molecule inhibitor of ROCK2. FDA has recently granted breakthrough therapy designation for KDO25 for treatment of chronic GVHD after failure of 2 or more line of therapy. For those who don't know, KDO25 is being tested for the treatment of idiopathic pulmonary fibrosis. Inhibiting ROCK2 may improve scarring and prolong survival in pulmonary fibrosis patients. Drug is produced by Cadmon Biopharmaceutical Company. With promising results before, investigators designed this Rockstar study. Patients receiving 2 to 5 lines of prior systemic therapy were prospectively randomized to once daily or twice daily dosing of KD025. Treatment allowed to continue until significant progression of chronic GVHD or unacceptable toxicity. Primary endpoint of study was overall response rate per NIH criteria. Slide summarizes the statistical analysis plan, and authors presented interim analysis 2 months after completion of enrollment. Data cutoff date was October 17, 2019. 12-month follow-up analysis is expected in fourth quarter of 2020. Patients were enrolled at 28 US sites listed here. Study fully enrolled, fully enrolled in less than 10 months. Here is the baseline characteristic. Severe chronic GVHD by NIH criteria in 2/3 of patients. More than half of the patient had 4 or more organ involvement. Prior ibrutinib is more than 1/3 of patients, not uncommon nowadays because ibrutinib is approved for chronic GVHD. Majority of patients were refractory to prior line of therapy. Median follow-up duration was 5 months. Slide summarizes the disposition at this time. Blue boxes at the bottom. At this time, 2/3 of patients in each cohort remain on active therapy. KD025 was extremely well tolerated, and I have myself witnessed this in several of our patients followed at Vanderbilt Long-Term Transplant Clinic. No apparent increased risk of infection. Infection is the main concern in most alternative systemic immunosuppressive medications we used to treat chronic GVHD, right? No increased risk of infection on KD025 and no CMV reactivation. Primary endpoint met at the interim analysis occurred 2 months after last patient was enrolled. 2/3 of the patient achieved CR or PR, and response expected to continue to increase slowly over time. As you have seen in a previous slide, more than 2/3 of patients were active on the therapy. You can see here, lower bound confidence interval is more than 50%, robust result. Slide in the forest plot summarizes the report response observed in all key subgroup. This is what we wanted to know, how they did in this subgroup. Responses were better for patients with less severe and short duration from diagnosis to study group. No differences stratified by number of organ involvement. 2/3 of patients responded in each group. No outcome difference by prior exposure to ibrutinib or ruxolitinib, Jakafi. Again, 2/3 of the patients responded in this group also. In summary, KD025 was well tolerated, achieved clinically meaningful outcome. To me, looks like we are going to have another approved drug for chronic GVHD soon. Another late breaking abstract, and this was a fantastic study. I thank Dr. Berdeha and his team for sharing this presentation. CAR-T1 is a phase 1b, phase 2 open label multi-center study evaluating safety efficacy of BCMA-targeted CAR-T therapy in patients with relapsed refractory multiple myeloma. Very important study for next few months. CAR-T1 has a 2 portion, like I said, phase 1 portion and phase 2 portion, phase 1b and phase 2. Primary objective of phase 1b portion is to consider safety and confirm the dosing. And primary objective of phase 2 portion is to study the efficacy. Key eligibility criteria summarized here. I'm not going to go into detail. Heavily pretreated population and exposed to almost all possible novel agents. Here on your left, you can see study design, standard CAR-T clinical trial study design, screening, pharesis, bridging therapy as needed, lymphodepleting chemotherapy with flu-cy, BCMA-targeted CAR-T therapy, post-infusion assessment and follow-up. Target CAR-T dose was 0.5 to 1 million CAR-T positive viable T cell per kilogram. Author presented phase 1b portion of the study, which had a median follow-up of at data cutoff at 6 months in a follow-up. Total 29 patients treated in phase 1 portion of the study. As you can see here, all patients were heavily pretreated, median prior therapy line 5. All patients were triple exposed, and most were triple refractory. 1/3 of patients were penta-refractory. All 28 of 29 patients were refractory to their last line of therapy. Slide shows safety data. Severe non-hematological adverse events were uncommon. Looking at the CRS and neurotoxicity is our main area of interest when we do CAR-T, right? 85% of patients had a grade 2 or less CRS, but only one patient had CRS grade 3 and one had prolonged dose-limiting toxicity, with CRS grade 4 turned into grade 5. Standard management of CRS, most important finding was very few patients developed neurotoxicity. Only 3 had grade 3 neurotoxicity. Only one had grade 3 neurotoxicity associated with CRS, and it was resolved. Look at this fascinating, very impressive slide. Very impressive response. 100% of patients achieved a reduction in paraprotein. A rare finding, 100% objective response rate. And look at this. 69% of patients had STR, almost all with stringent CR. CR plus VGPR in 86% of patients. Overall, response rate and depth of response were independent of BCMA expression on multiple myeloma cell line at baseline. Median time to first response in most cases, most CR cases was 1 month. So most patients develop CR in a month. 27 of 29 patients are progression-free at 6-month follow-up. Responses deepens over time. You can see here many patients, you can follow the color pattern. Many patients in stable disease, gray, PR, you can see the color green, VGPR, violet color, later went on to achieve CR or stringent CR, blue color, ongoing. And now when we discuss novel study, first question comes in our mind is, what about MRD? That's the main question people ask. Look at the result. 100% of evaluable patients were MRD negative at day 28. Slide summarizes the J&J 4525 CAR-T expansion and persistent. Peak expansion was 30 days after infusion. In 18 of 28 patients, number of CAR-T cells were less than 2 cells per microliter at 3-month follow-up. Now, important finding is CAR-T cell disappeared, but that does not mean detectability of CAR-T cell did not appear to correlate with continuing deepening of response. Very important. In summary, J&J 4528 CAR-T therapy directed against BCMA in patients with relapsed refractory myeloma is very effective. And let me briefly mention about other myeloma CAR-T product. Bluebird product called ID cell. This has also a long name which I can't pronounce. IDE cell, BB2121 CAR-T therapy for relapsed refractory myeloma. Data were presented at the ASH meeting. Again, very impressive outcome. Our last abstract of the session, safety and efficacy result of Transcend NHL 001 multi-center phase 1 study of Lysocell in relapsed refractory large B-cell lymphoma. Lysocell is a Juno product, then bought by Celgene and now bought by BMS. FDA granted priority review of liso-cel in relapsed refractory large B-cell lymphoma just before the TCT meeting on February 13, 2020. Liso-cel is a 4-1BB CAR-T product administered at target dose of CD8 positive and CD4 positive CAR-T. Author presented Transcend NHL 01 data in large B-cell lymphoma. Studied included all adults with advanced B-cell lymphoma. Important aspect of this study was patient with mild or moderate organ dysfunctions allowed in the study, and also central CNS involves were eligible, like we mentioned before. Liso-cel was given at one of three doses, 50, 100, and 150 viable CAR-Ts per 10 multiply 10 viable CAR-T cell. Majority of patients received dose level 2, and it was chosen for the dose confirmation. Standard primary and secondary endpoint, toxicity and efficacy. No need to go into the details. And this is the result. Only in two patient product could not be manufactured, which is very good compared to other product we discussed, especially Kymriah product. Not bad from this large TCT. 25 patients received non-confronting product, generally viability or cell dose less than 80%. As I mentioned before, 80% is a target dose in most centers, but okay in most cases, even if we go a little less than 80%. Lots of older patients, 42% were age 64 or higher. Some patients received liso-cel as an outpatient. Less than half of the patients had any grade CRS or neurological event. But that's not important. Important thing is only 2% of patients had grade 3 or higher CRS, and 10% with grade 3 or higher neurological event. No grade 5 events. Much better than many other CAR-T products reviewed for lymphoma today. Did you see another important finding in this slide? CRS and neurological events were late in onset. Median time to CRS 7 days, neurological event median time 7 days, meaning we can keep patients outpatient for a long time after CAR-T infusion. Median overall response rate was 73%, CR 53%. Median duration response was not reached at 12-month follow-up. Median duration of response for patient in CR was not reached. You can see here both upper bound and lower bound confidence interval was not reached. In conclusion, liso-cel showed an excellent durable clinical activity in relapsed refractory large B-cell lymphoma subgroup. Incidence of CRS and neurological events were very low, and most important, late onset, allowing for outpatient administration. I can see here there are still several, almost 2/3 of attendees are on board. I will briefly add a few more slides. This is very important because it looks like you guys are still interested in if I'm going to continue. Just last couple of slides. CAR-T toxicity grading system. Very quickly, reviewing this educational session presented by Dr. Nilopu, our well-wisher, and I thank him for sharing this presentation slide. You know, what's our big problem? Variety of IACH CAR-T toxicity grading system. Let me start with this case scenario. 54-year-old patient with diffuse large B-cell lymphoma developed hypotension requiring low-dose vasopressor for 2 days after CAR-T therapy. How CRS will be graded? It will be graded grade 2 in 2014 Lee or CAR-Tox cell. It will be graded 3 on Penn or Memorial scale. It will be graded 4 on CTCA-4 scale. So I'm sure you all agree that assessment and grading of these toxicities are very considerably across clinical trial and across institution, making it difficult to compare the safety of different products, making it difficult to develop optimal strategy for management of this toxicity. Moreover, some of the aspect of the grading system can be challenging to implement across the center. Like we have to have something not only great, but also implemented by all. Therefore, in an effort to harmonize the definition and grading system for CRS neurotoxicity, expert in the field met in June 2018 at meeting supported by ASTCT, developed the ASTCT consensus grading for CRS neurotoxicity associated with IEC. Article was published in April 2019. IAC grading of CRS and icons available in F form, supported by both iPhone and Android platform. App was developed by Dr. Paralis and team, also team member from ASTCT practice guidelines and educational committee. There is another CAR-Talks app published by the team at MD Anderson, Sherry, and Nilupu and other institutional members. There are still some limitation of this ASTCT grading. Toxicity not addressed in ASTCT grading are prolonged cytopenia. We discussed today. There are patients who die beyond 1 month after CAR-T from infection due to persistent pancytopenia. Hemophagocytosis syndrome, macrophage activation syndrome, B-cell aplasia, hypogammaglobulinemia, GVHD after CAR-T is still not graded in ASTCT grading. Work is in progress at ASTCT, and also ASH is developing consensus CRS and ICON management guideline will be coming out soon. I'm sure ABMT colleagues are also working on this direction to help our patients across the globe. And I thank you very much. Special thanks to our friends, colleagues, and well-wishers who provided their slide to discuss their data with us today. And both part 1 and part 2 TCT summary webinar will be available on IACH website and also direct link to YouTube video. Goal is to share the information to help our patient, and I thank you all for your patience. I'll be happy to answer any question. I think we still have lots of people online.